Our next speaker is Professor Claudio Babiloni from Rome, Italy. He's a professor of physiology at the medical faculty of Sapienza University. His main scientific interest is focused on the neurophysiology of Alzheimer's disease and other neurodegenerative dementing disorders by the study of resting state EEG rhythms, both from basic science all the way to clinical trials. He's the leader of the IFCN EEG research work group, and he will present the guidelines on spatial and frequency analysis of EEG rhythms. Claudio, please.
Thanks a lot, Blas, for inviting me to this fantastic conference on guidelines in clinical neurophysiology. The title of my presentation is about the International Federation of Clinical Guidelines of the spatial and frequency analysis of resting state EEG rhythms. Hans Berger, an Austrian psychiatrist, recorded the resting state, eyes closed and eyes open EEG rhythms in 1928. It showed an ample EEG oscillations from 8 to 13 Hz on posterior areas and called alpha rhythms. These rhythms were reduced in amplitude during the eyes opening and mental calculation. And you can see the appearance of higher frequencies from beta to gamma. And the EEG rhythms are generated and induced by ascending activating system and thalamocortical interactions. You see here the reticular formation and the serotonin, dopamine, noradrenaline components, and the basal for brain using projections and acetylcholine to modulate the cortical activation. And of course, the important role of thalamocortical interactions in modulating the generators of EEG rhythms in the cerebral cortex. The spatial and frequency analysis of resting state EEG rhythms can produce biomarkers for clinical neurophysiology research. And these biomarkers can be used to disentangle patients and to form neurophysiological models of the diseases from symptoms onset to follow up. The International Federation of Clinical Neurophysiology in 1999 asked a smart work group to provide recommendations about the procedure for the recording and analysis of resting state EEG rhythms. The work group accepted electrode montages lower than 19 electrodes and suggested the recording of EEG data for at least 1 minute, and accepted digital recording storage and the use of source density derivations in order to enhance the spatial details of resting state EEG activity. Finally, they suggested the use of more than one electrode reference for the topographic mapping of resting state EEG rhythms. You can see the publication of these guidelines and the names of the smart scientists cooperating for this for that paper. And after 20 years, there are new challenges for the recording and analysis of resting state EEG rhythms. You can see that there are discussions about the proper resting state condition, about the use of high-resolution EEG in clinical neurophysiology, the frequency bands, and the individual basis for the choice of frequency limits, and the choice between analysis at sensor or source level And the kind of techniques for studying functional connectivity from EEG data, and the debate about the linear or non-linear features of resting state EEG. To tackle these questions, International Federation of Clinical Neurophysiology asked a work group, and you can see the names of the scientists participating to this editorial challenge. And the work group suggested that the proper resting state condition depends on the neurophysiological mechanism that you want to study in your patients. For example, resting state eyes closed for 3-5 minutes and eyes open for 3-5 minutes are useful to investigate the maintenance of a stable vigilance. And quick changes of eyes closed and eyes open are useful to investigate the switching of a vigilance level. And the use of long recordings, more than 30 minutes of resting state, eyes closed, are useful to investigate the progressive transitions among vigilance level from quiet wakefulness to sleep. The work group suggested that the proper electrode montage depends on the spatial and clinical issues in your clinical research. For example, the 10-20 montage system plus additional 6 electrodes in the left of the figure is useful for the topographical analysis of resting state EEG rhythms. The use of 10-10 system up to 81 electrodes is useful to study the cortical sources of EEG rhythms. And for the source location of epileptiform activity, the work group suggested the use of up to 256 electrodes of a 10-5 system. The EEG signal is complex, but can be decomposed in elementary oscillations at different frequencies. And this allows the individuation of individual alpha frequency peak, the frequency showing the maximum power in the range of alpha rhythms. And the individual alpha frequency peak can be used as a benchmark to define the individual delta, theta, and alpha frequency bands. And you can see that the magnitude of these oscillations can be represented in the EEG power spectrum. Of course, you can also use fixed frequency bands according to the tradition of resting state EEG. But you can see that different guidelines from International Federation of Clinical Neurophysiology and Pharmaco-EEG Society propose different frequency bands for alpha, beta, and gamma. So it's an open issue, the choice of the proper fixed frequency bands for clinical neurophysiology research. This is an open issue. The work group addressed also the issue of head volume conduction effects, challenging the spatial analysis of resting state EEG rhythms. You can see in the picture the two examples of a head volume conduction effect. On the right side, you see an EEG signal recorded in the electrode B, but the sources is not underlying the electrode B, but it's located in the tangential sources far from the electrode. Another example of head volume conduction effects are in the left side of the picture. You can see in the electrode A and B two signals which are generated by the same source, radial source halfway. So if you apply techniques for the analysis of interdependence of EEG signals at electrode base, you can see a coherence of these signals but no source connectivity underlying. To tackle the head volume conduction effects, it's possible the use of source estimation techniques. In the left side of the picture, you can see an example of a procedure solving the inverse problem of EEG. From scalp EEG activity, you can estimate the underlying cortical sources using a model of head volume conduction effects. And you can also use other independent techniques to estimate the radial current density, not using complex models of the brain and sources. The work group proposed that the use of two independent techniques for source estimation from resting state EEG rims allow you to have reliable solutions for clinical neurophysiology research. The work group also addressed the important chapter of the functional connectivity from resting state EEG rims. And of course, you can address this analysis with linear and non-linear techniques. You see an example of in the upper part of the picture of linear interrelatedness of two musicians in phase each other and producing a performance in a linear way. And in the lower part of the picture, you can see an example of a non-linear interrelatedness in which you have two musicians that provide sounds not using the same phase of activity, but the sounds are harmonize each other in the performance. And this is similar to what you can find in the EEG sources of resting state EEG rims. Of course, it's important to take into account both linear interrelatedness and non-linear interrelatedness when you study the EEG source connectivity. In the right side of the picture, you can see an example of analysis at the sensor level and an example of analysis at the source level. In this slide, you see an example of synchronization Lahai-Klude technique introduced by Professor Stam, able to capture both linear and non-linear interrelatedness of EEG signals at electrode pairs. You see that from EEG data recorded from frontal electrodes, for example, you can measure amplitude in the red time and blue time, and after a short period, again red time and blue time, and so on. And you can represent this in a multi-domain space. For example, in this simple case, two domains, the red domain and the blue domain, and you can use the amplitude measure to have dots in this space. And repeating the operation several times, you have a trajectory. And you can repeat the analysis in another electrode and obtain other trajectory, and another trajectory. And you can estimate the interrelatedness between the two trajectories to have a measure of synchronization likelihood of EEG signal in electrode pairs. You can also, of course, estimate the sources of the connectivity between the sources, but you are challenged by head volume conduction effects. You can see two examples of these challenges. In the common drive condition, you have the source C coupled with the source B and the source C coupled with the source A. When you apply the techniques for estimating the interrelatedness of EEG signals at electrode pairs, you can capture this relationship between C and B. You can obtain a fake interrelatedness from B and A. You don't have this in the source space. And the same is true for the cascade flow phenomenon. If you have the source C coupled with the source B, and the source B coupled with the source A, you can capture these at the electrode level with the techniques similar to synchronization ligand, but you can also have a fake interrelatedness between EEG signals, not related to source connectivity. So the work group concluded that the functional connectivity EEG techniques need more validation before a reliable use in clinical trials. In conclusion, linear frequency and topographical source analysis of EEG rims are major for clinical research. Nonlinear frequency and connectivity analysis of EEG rims may require consensus before a full use in clinical research and clinical trials. The work group concluded that there is the need for international scientific initiatives to address main challenges in the use of resting state EEG rims in clinical neurophysiology. For example, to better understand the use of linear versus non-linear measurements, the issue of fixed frequency bands, and the issue of EEG functional connectivity and the use of graph theory markers, very promising in this field. And so we recommend that the future position and white papers on the use of resting state EEG in clinical research and clinical practice after these international consensus initiatives. Thanks for your attention. Here is my group in Sapienza University in Rome. They are the main contributors of this presentation, and these are the European funds supporting this research. Thank you.
Thank you very much, Professor Babiloni. Maybe while we are waiting for any questions to come in, where do you see possible clinical applications of connectivity analysis? Do you think that this will eventually come into a regular clinical practice diagnostics?
Okay, this is really important. Our work group work on the use of resting state EEG rims in clinical research, which means, for example, intervention clinical trials, or the multimodal EEG, magnetic resonance imaging, and PET combination of measures to understand the model, the neurophysiological and neurobiological model of the diseases. We didn't address the practical use in the management of patients in the clinical practice. There is an action in progress to look for a consensus about what kind of quantitative advanced EEG techniques can be used in the clinical practice, including the functional connectivity techniques, source analysis techniques. However, we know that several smart groups in the world use EEG source connectivity and estimation of EEG sources from resting state EEG data for several clinical applications, such as the management, the workup of epilepsy patients to localize the sources of epileptiform activity and enrich the planning of neurosurgery in epilepsy patients resistant to pharmacological therapy. And other groups are using, for example, resting state EEG RINs and the quantitative measures of spectral analysis in the field of Lewy body dementia. And the recent guidelines on the diagnostic criteria of Lewy body dementia includes as a supportive biomarker the delta and theta power from resting state AG eyes closed. So there is an increasing interest also for the use of these techniques in the clinical practice.
Thank you. What about, do you think that the connectivity analysis, are they just too complicated for a simple clinician to grasp and to understand?
Yeah, absolutely. This is our work group found a big heterogeneity of the procedures to estimate functional connectivity from resting state EEG. We found that there are several procedures, several techniques to measure EEG functional connectivity, several procedures to have statistical results and at the individual level. So you are right that there is no consensus up to date to offer clinicians clear procedures for decision making in clinical practice. You are perfectly right, and we are working to reach some consensus between stakeholders and main groups in the field with the International Federation of Clinical Neurophysiology.
And what about the automated types of analysis, machine learning algorithm and so on? Do you think that we should expect things to develop into the direction where we will only press the button to get the results?
Yeah. There is an increasing interest in the use of machine learning's artificial neural networks to get a lot of EEG biomarkers at the same time and to provide a single information clinically relevant. The main weakness of this approach is that, again, we don't have consensus about the standards to separate the preliminary selection of the EEG features and then the use for the training phase of the machine learning procedures or artificial neural network or supervector machines, and then the final use in the testing phase, providing you the clinically relevant information. However, we agree on the fact that any validation of these procedures should be based on the use of independent databases of EEG data in order to test if a trained learning machine is able to provide diagnostic information or prognostic information with a high accuracy, even in an independent database.
Yeah, thank you very much. See if there are any questions. No, I don't think we had any other questions. Okay, thank you, Professor Babiloni. Thank you very much for your presentation, for being with us, and hope to see you in Ljubljana at some time.
Yes, it has. I was very happy to work with this work group to produce the guidelines for International Federation of Clinical Neurophysiology. But it has been fantastic to have this opportunity to disseminate this work with you and the other colleagues of the Slovenian but also international, because I see that you have a lot of participants now. Thank you very much.
Thank you. Bye-bye.
Bye.